Unlike the innate defenses, adaptive defenses must be primed by initial exposure to the specific pathogen before being able to mount a defense. That means that we are not immediately able to fight off that particular pathogen. Instead, we must learn how to fight that pathogen. However, the adaptive defenses do have the benefits of specificity, that is, recognizing and destroying particular pathogens, like particular viruses, and as viruses mutate and change, our adaptive defenses can learn to fight this new form of the virus. They're also systemic, that is, that the immunity is not restricted to the site of the initial infection, like we see for most of the innate kinds of defenses. And adaptive defenses show a type of memory. That means that a second exposure to the same pathogen leads to a rapid, massive response. For example, most people who had the chicken pox once in their lifetime don't get sick from that same virus again because the memory of their adaptive defenses allows them to initiate a very rapid, massive response to prevent a spreading of the chicken pox virus. The adaptive defenses rely on two interconnected arms or sides. The first is humoral immunity. Humoral immunity uses antibodies circulating in the body's humors to help identify pathogens that need to be destroyed. And then cellular immunity. Cellular immunity includes lymphocytes that act either directly in defense or increase inflammation, meaning we have cells acting against other cells. That's why it is a cellular form of immunity. Two important groups of chemicals make the adaptive response possible. These are antigens and antibodies. Antigens are substances that mobilize the adaptive defense responses. They are intruders, or non-self particles. Antigens are the indication of pathogens being present in the system. Antibodies, on the other hand, are produced by the immune system. Antibodies are produced specifically by B cells of the immune system and can stick to antigens. They are part of the defense against antigens and pathogens. Antigens can be either complete or incomplete. Incomplete antigens also go by the name of haptens. Complete antigens have two functional properties. The first is immunogenicity. That means they can cause a specific lymphocyte to proliferate and try to destroy that pathogen. And they exhibit reactivity, the ability to react with activated lymphocytes and antibodies. Nearly all foreign molecules can be complete antigens, including proteins, large polysaccharides, some large lipids, nucleic acids, even entire cells like bacterial cells. Incomplete antigens, or haptens, are too small to cause an immunogenic response on their own. But they can leak to proteins within the body that are large enough to be recognized. This can include small peptides, small nucleotides, and many different kinds of hormones. Ability of a molecule to react to an antigen depends on its size and complexity. Only certain parts of an antigen are immunogenic and cause an immunological response. These parts are referred to as the antigenic determinants. This is where antibodies will bind when mediating the immune response. Most naturally occurring antigens have multiple antigenic determinants, multiple places where antibodies combined on their surface. Different lymphocytes will recognize different antigenic determinants. Different lymphocytes will use different antibodies as part of the immune response, each of which will only recognize specific antigenic determinants. A single antigen, like a bacterial cell for example, can mobilize multiple lymphocytes simultaneously because each lymphocyte might respond to a different antigenic determinant on that antigen. The more lymphocytes that get activated, the larger the immune response. To help with that, let's take a look at a possible antigen. Here, we can see one large antigen with three distinct binding sites, three distinct antigenic determinants. Because it has three distinct antigenic determinants, three different antibodies can attach to this one antigen. That could cause a relatively large immune response because we have multiple antibodies, multiple B cells, and multiple T cells that can respond to the different antigenic determinants on this one antigen. This helps us understand why pollen can cause such a large immunological response in folks who are allergic to that pollen. Every pollen grain is covered in a lot of different types of antigenic determinants. We can see multiple three-dimensional structures, each of which would cause a different kind of antibody or a different kind of immune cell to respond to the presence of that pollen. The more immune cells, the more antibodies that are attaching to this antigen, the larger our immune response will be. A series of glycoproteins, called MHCs, cover the surfaces of all of our cells. These are your self-antigens, letting your immune system know that those cells do belong there. Your MHCs should not elicit an immunological response from your own immune system. But because your cells are covered in these MHC kinds of antigens, they would be viewed as non-self by the immune system of other people. This is what forms the basis for the rejection of blood transfusions and organ transplants. That is, a mismatch of these antigenic determinants. Those that your cells should be producing on their own are different than those produced by other people. These MHCs are coded for by multiple genes and can result in millions of MHC combinations, meaning that it is unlikely that any two people, who are non-identical twins at least, have the same combination of genes and the same combination of MHC antigens. That's why it's so hard to find a correct donor to a person who needs an organ transplant. We're trying to find the closest match of these millions of possible combinations of MHC antigens.